Ladies and gentlemen, friends and colleagues, thank you for joining us today for the 2018 Marino Xanthos Memorial Lecture. The lecture was organized by family and friends of the late Marino Xanthos, who passed away in summer 2013. By that time, Professor Xanthos had served at NJIT for 18 years, having worked earlier at Martin Marietta Resources International and at the Stevens Institute of Technology. Among his positions here at NJIT, and in addition to his role as Professor of Chemical Engineering, he was Director of Polymer Engineering of the Polymer Engineering Center at NJIT, Director of the Center of Processing of Plastics Packaging, Chairperson of the Executive Committee of the Materials Research Council, Senior Technical Advisor to the Polymer Processing Institute at NJIT, and Associate Provost for Graduate Studies. Professor Xanthos was internationally recognized, an internationally recognized expert and researcher in the fields of reactive polymer processing, polymer extrusion foaming, and polymer nanocomposites. He was also a strong advocate of plastics sustainability issues. One of his last papers, and one of which he obviously was very proud, was published in August 2012 in a special issue of Science devoted to working with waste. The title of this issue shows a photograph of bales of compressed aluminum cans destined for recycling. The Marino Xanthos article in this issue was titled Recycle of the number 5 polymer. It deals with recycling of polypropylene, PP, and I'm showing you here a figure from this Science article. PP is a thermoplastic polymer used in a wide variety of applications, including packaging and labeling, textiles, applications such as ropes, thermal underwear and carpets, stationery, plastic parts, and reusable containers, laboratory equipment, loudspeakers, automotive components, and polymer banknotes. The article discussed the factors contributing to PP recycling rates, which include collection and separation efficiency, processing chemistry, and market dynamics for the products derived from the recyclates. Let me also mention the 2 publications of Marino Xanthos, which have garnered the highest number of citations from among his studies. Here is an image of the cover of a book he edited and published in 1992, Reactive Extrusion, Principles and Practice. It has by now quoted close to 470 times. About 70 of those have been made during the last 12 months since we met here for the Xanthos lecture last time. A second book, Functional Fillers for Plastics, was published by Wiley and had a second edition in 2009. About half of it was written by Professor Xanthos, and it has been cited in the literature close to 370 times so far. I did a quick check of what happened since 2017. You see 52 citations in a variety of language. Most of them are, of course, in English, but there are some in other languages as well, and you can see the list of publications where this book has been cited. Today's lecture follows a technical session in memory of Professor Xanthos. It was held in the 2014 Society of Plastics Engineers Annual Technical Conference in Las Vegas. In 2015, we had a memorial lecture held at NJIT. It was given by Professor Morton Den of the Benjamin Levitch Institute at the City College of New York. The 2016 memorial lecture was given by Professor Thomas Russell from the University of Massachusetts Amherst, and the 2017 memorial lecture, and this was the advertisement to it, was given by David Kaplan from the Department of Biomedical Engineers at Tufts University. It is the intent and plan of NJIT to organize and present this lecture on areas close to the professional focus of Professor Xanthos in perpetuity. And I'm pleased to acknowledge the work of the committee in charge of this task, which is chaired by Professor Piero Armenante, and whose members are our colleagues Lisa Axe, Cheryl Baker, Rosalind Manning, Kamalesh Sirkar, Angela Vega-Irvin, Trevor Tyson, Cynthia Voss, and Carolina Yanez. I'm pleased to invite now for incoming remarks on behalf of NJIT's President and Provost, Professor Basil Baltzis. Professor Baltzis was a colleague of Professor Xanthos, and he is a professor of chemical engineering, as well as Senior Vice Provost of Academic Affairs and Student Services at NJIT. Professor Baltzis.
Good afternoon, and welcome to this event. On behalf of President Bloom, who is traveling in China on business, and the Provost, Dr. Fadi Dik, who had to be at the faculty meeting, the Institute faculty meeting, which is taking place right now, welcome and thank you for attending this event. Both the President, the Provost, and I had the opportunity from different positions to work with Marino. I had the opportunity and the good fortune to meet him ever since he came to NJIT from Stevens, and he was a person of very sharp intellect, a person who, despite some physical disability, showed to everyone that one can have a great life, can be compassionate, can be passionate, without allowing physical issues to intervene and affect his or her life. He was extremely student-focused. He was spending an enormous amount of time with all his advisees. I remember on the third floor of Tiernan Hall, where both he and I had an office, even late at night, he was spending time with his graduate students in particular. He was always the person who would volunteer to serve on different committees, and he would always have some nice contributions to make and some interesting ideas to contemplate. For those of us who really met and worked with him, sometimes it feels as if it was only yesterday he was with us, and sometimes it feels as if a very long time has elapsed. It is hard to believe that it has been already 5 years. I'm very happy that his children, his sons, Dimitri and Haris, are here with us today, and it is because of their initial first generous contribution that the fund was established to have, in memory of their father, this lecture series annually. And once again, I would like to thank them, and I would also like to thank the organizing committee, the Dean of Engineering for supporting this event, and of course, our speaker today. And once again, I apologize for both the President and the Provost not being able to be with us today. Thank you.
In addition to thanking the Xanthos family for its support, I wanted to remind everybody that Professor Xanthos was a popular teacher and student mentor. He was appreciated, and he is now remembered by his many students at all levels of education. We welcome today some of his advised graduate students in the audience. They have joined us today for this lecture, and I'd like to thank them in particular, as well as other advised students of Professor Xanthos, for assisting with the annual memorial lecture through direct support and through attendance at this annual event. Today's speaker and lecturer will be presented by Professor Piero Armenante. Professor Armenante is a distinguished professor of chemical engineering at NJIT, and he also serves as director of the NJIT graduate program in pharmaceutical engineering. He is the chair of the Marino Xanthos Memorial Lecture Committee. Professor Armenante.
Thank you, Dean Kam. So it is always a pleasure for me to be here for this particular series of lectures. I look forward every year to share this experience with you after a lot of work that we put into the effort of selecting the candidate, contacting everybody, assembling the team, and making sure that we present NJIT the way it is. In other words, its best aspect. And in fact, as you will we'll hear in a moment, Professor Wooley had the chance also of exploring the campus and talking to a number of faculty members. However, before introducing a lecture today, I'd like to at least briefly remember Marino. Marino Xanthos, as was mentioned before, was a colleague of mine, but a colleague of mine that I work with. And there was, we were in different fields, but still, we collaborated on a number of projects. And we shared students. In fact, one of my former students that completed his master with me worked with Marino to go on for his PhD. And too bad he's in China now, otherwise, China on a business trip, because otherwise we'll be here to remember Marino. I had the fortune of working with Marino as one of the last things that he did. So we put together a program that actually was in the beginning of its existence in materials with special focus on processing of pharmaceutical materials. And this was the last thing that we did together, because very soon after that, he passed away. So I'm sorry that we could not have completed this project the way we wanted. But nevertheless, I, of course, very good memory of working with Marino. I remember his competence. I was invited to be on a committee of his advisee, and I did the same with mine. And in addition to his scientific contribution and excellence in teaching, I always have this personal image of him, which you can somewhat see in the picture which is in the back of your brochure, in which he was always looking, especially was approving things, was looking with some kind of ironic, kind of dry smile, kind of saying, yeah, this is good, but never saying this too explicitly. So that's the image that I have of Marino. So I, of course, am going to introduce today's lecturer, and I will do this in a moment. But before doing so, I'm glad to see that some of his children are here. Maybe one of you wants to say something remembering Marino. Would you like to say a couple of words? Sure. Please.
Hi, I'm Dimitri, one of Marino's son. Actually, we just, on behalf of the family, wanted to thank Professor Armenante and the organizing committee for once again organizing very well this lecture for the 4th year in a row, and bringing here a distinguished speaker and scholar. I also wanted to thank NJIT for their continued support of this lecture, and of course, I wanted to thank Karen Wooley for coming over here. And I'm very much looking forward to her lecture, which I think does align quite well with many of Marino's interests. I'm certain that Marino would be happy that this lecture series is still taking place. He was always supportive of knowledge sharing, breaking boundaries, and collaborations. And we hope that this lecture series can bring about further benefits as such for NJIT and for the wider scientific and chemical engineering community.
Thank you.
Yeah.
So, as I said, I I'm very proud to have the opportunity to chair the committee to honor Marino through this kind of series of lecture, which is becoming more important as time goes by. So I'd like to acknowledge, of course, all the people that did the work, and so in particular, beside the committee that was mentioned several times before, I'd like to acknowledge the contribution of Rose Manning and Sharon Baker, who were absolutely instrumental in assembling the little details that are critical to making sure that this works very well. Now, let's move on to the introduction of our speaker for the lecture for today. Dr. Karen Wooley. You have in front of you her scientific accomplishments, but actually I noticed that at least since I've been running this, the speakers that we were able to bring here to NJIT for these important lectures don't only have a super strong scientific background, but also have a personality. So I had the chance to interact with Karen yesterday, and it was really fun in a way. You know, we went out for dinner in the Newark Ironbound section, and after just a few minutes, we were all, you know, a bunch of people talking about serious stuff, but also talking about light components. I mean, life experiences, children, place of living, whatever. So wine, you know, she's from Oregon. She seems to be a very good connoisseur of especially Oregon wine. So at the end of the evening, you know, we were all in very good spirit. And in fact, I drove her back to the Hilton Hotel where she's staying. And you know, yesterday was Mega Million, the lottery. So, okay. So she said, I need to get a ticket. So we were coming back from the restaurant, and we actually went through downtown Newark looking for a place where we could buy tickets. So we stopped, and she said, you must buy tickets too. I said, no, come on, I never played in my life, but she said, no, you have to. So I had, you know, okay, invested $10 in lottery tickets. We took pictures with the tickets in our hand. So this is the kind of stuff that I'm referring to. It's not just the scientific proficiency or excellent quality of our speaker, but also the personal touch. So of course I need to explain to my wife why I had 5 tickets, because I never bought any tickets in my life. So she said, what happened? So I tried to say, well, this was for you as a present, but then I had to confess that actually it was downtown Newark with a crazy lady that forced me to buy tickets. At any rate, so now on the more serious stuff. I don't particularly like to read all the details which you can see yourself. I like just to point at a couple of things here and there. But Karen Wooley is the W.T. Doherty Welch Chair in Chemistry and University Distinguished Professor and Presidential Impact Fellow at Texas A&M, which by itself obviously tells you the stature of Karen. She also has appointments in other departments, chemistry, chemical engineering, material science, and so on. And she serves as the director of the Laboratory of Synthetic Biology Interaction. And she has a number, of course, of academic degrees, including her PhD from Cornell. And she had also distinguished career at different universities, where she served in different capacities. Now, she has a variety of interests, especially in novel synthetic strategies. And as a result of her work in a variety of fields, again, I don't want to bore you with just reading all the details of what she's doing, because she's involved in many different things. She has been acknowledged as a leader in her field, and so at least I'd like to mention that she received the Academic Chemical Society Award in Polymer Chemistry, the Royal Society Chemistry Centennial Prize. She's a fellow of the Royal Society of Chemistry. You need to be, elected to that. She's an honorary fellow of the Chinese Chemical Society. She's a fellow of the American Academy of Arts and Science, and many other distinguished records, but also like to point out that she is an associate editor for a very prestigious journal, which is the Journal of the American Chemical Society. I think it's probably #1 publication in the area. So again, you can read the details in the brochure. And at this point, I think, without any further ado, I think it's my time to introduce now our lecture for the 4th edition of this series of lectures. So please welcome and Wooley.
So I just have to say thank you so much to Piero for many things. I'd like to thank you and your committee for selecting me for this honor. I'd like to thank you for the introduction. I've never been introduced as a crazy lady, although I think everyone in my world knows that I'm a crazy lady, so he recognized that in only one evening. We did have quite a fun time. The last slide of the presentation, you'll get to see the highlight of the ticket buying last night. But let's go on a more serious note. I would like to thank everyone with whom I've met today. I enjoyed it very much. I've been extremely impressed with the facilities and the people here. I'd also like to thank Dimitri and Harris for the dinner last evening. It was a pleasure to get to meet you, and I look forward to following up with you in the future, maybe as we compete against Abraxane, for instance, in our work. But it's been really a delightful opportunity. And I think what's really important is that what brought me here is the opportunity to remember a very special person. And this special person, I unfortunately was not able to meet, but I can recognize how great this man was. And I'm just going to show three pieces of work of his that relates to how he really has been making accomplishments much ahead of their time. And the first is one that was already shown, functional fillers for plastics. The need to put fillers into plastics is something that is still of current high importance. And some of the work I'll show you today involves loading polymer plastic materials with fillers to gain, in this particular example, magnetic responsivity by using inorganic particles as those filler materials into the plastics. The second is another book that was also shown earlier, Reactive Extrusion. And the importance of being able to transform materials as they're being processed. And that gives a high degree of power in making one particular material and then transforming it during the processing procedures. And then the third is this book on how to manage plastic waste. And again, this need to deal with waste materials and recycling is something that is becoming of paramount importance. And that's a real key point of the work that I'm going to talk with you about today. So what I'm going to discuss is this interest in developing synthetic methodologies by which to take small molecules and link them together into polymer materials, where those polymer materials are designed to have function to be used in various applications, but with an attention also to sustainability. And sustainability in a couple of different aspects. Sustainability in the function that the polymer materials exhibit, but also sustainability in how we make those materials and what resources we use and feedstocks we use to do so. So one of the first applications that we'll discuss is this need to take care of the environment, and take care of the environment in a case where there's been some damage to the environment. And the damage that's illustrated in this image is a oil spill where it's difficult to see, but you maybe can see the sheen that's present floating on the surface of the water over in this region. You might also be able to see that there's a bulk crude oil beneath the surface over in this region. So these populations of oil after a spill are difficult to clean up. One, because it's beneath the surface, and the second, because it's a thin layer on the surface. And what we've been doing is developing nanoparticles that can address this need and clean up these hazards from the environment. And these are, again, based upon putting fillers into polymer materials, where those fillers are inorganic nanoparticles. They're illustrated here as black spheres, where they're within a framework of an organic polymer material that can sequester crude oil through hydrophobic interactions to capture it within the core and then use a magnetic response to recover the materials. So I'll explain to you a lot about how we designed those, synthesized them, and then tested them. And then I'll proceed to how we began building those with just this interest in making functional polymer materials. again, to complete this important task, but not really being concerned about what building blocks we were using to make those materials. And then I'll transition into how we've become more keenly aware of the need to build into our functional polymer materials an end life and be able to take them back down, recycle them, break them down. And in that process, then we're using glucose and other sugars and other natural products as small molecules to build the polymers, to make the nanostructures to complete the particular functional application. We'll take a little bit of a sideline, if I have time, to discuss some other natural products that we're working on. These are polyphenolic-based natural products. These are meant to produce materials that can have physical and mechanical properties that can rival those of engineering plastics, to replace things like bisphenol A polycarbonate. Okay, so that's the overall outline for the presentation. So let's get started on the first topic, and that is cleaning up crude oil after it's spilled in the environment. So the typical cleanup procedures that are used are macro scale. For instance, after a spill has occurred, one of the first things that might be done is to put out booms that can prevent the oil from drifting downstream or migrating and contaminating other areas. And then after the oil is contained, then pumps can be brought in to withdraw the oil from the surface. And those procedures are fairly efficient, but what they fail to do, again, is to address this oil that might be beneath the surface or the thin layer that remains after these macro-scale removal processes have been conducted. So for the residual sheen, there are techniques used to, again, absorb that oil as well. And these are the use of things like polyethylene or polypropylene mats that can be laid out on the surface of the water. They're allowed to sit in place for a period of time to absorb the oil, and then they're bagged up and hauled away. So there are several problems with this approach. One of them is that these mats are only being used on one face, and they're floating on the surface. And then the other is that they're rather slow in taking up the oil, and it's a laborious process. You can see all of the workers in the background that had to lay out these mats, wait for them to absorb the oil, and then pick them up and haul them away. Okay, so we thought, well, there might be a nanotechnological solution to this. We thought if there's a contaminated water system, then we could put out into that aqueous system a dispersant. And this project started after the Deepwater Horizon oil spill had occurred in the Gulf of Mexico back in 2010. And one of the techniques to deal with the vast amount of oil that couldn't be boomed and pumped was to put out dispersants. And my thought was, if there's a polluted environment and then we're putting out surfactant molecules to disperse the residual oil, we're adding pollution after pollution, and it's maybe not the best solution. And instead, if we could put out those dispersants in such a way as they could capture the crude oil by sequestration, as dispersants would do by their hydrophobic core interacting with the hydrophobic hydrocarbons of the crude oil, then by having magnetic responsivity, then a magnet could be used to retrieve those materials, collect them, and if the oil could be extracted, then they might be able to be recycled and reused. So that was the overall approach, and we designed two different kinds of hybrid materials to do this. One of them is illustrated, as I've shown previously, where there's a polymer framework into which are embedded inorganic magnetic nanoparticles. The other, instead, has inorganic magnetic nanoparticles as central foci onto which are coupled organic polymer particles in sort of what we call a network approach. And these are called magnetic hybrid networks, whereas these are magnetic shell cross-linked Knedel-like nanoparticles. And Knedel is a Polish word for dumpling. So these have a core shell morphology, whereas these are more like a network structure. And each of these is accessed by a similar synthetic route. We start from small molecules and connect them together into polymers, where those polymers are programmed to undergo supramolecular assembly when they're placed into water to form multimolecular micellar aggregates. And then we can conduct, say, reactions to stabilize those assemblies. So that can give something that's called a shell cross-linked Knedel-like nanoparticle. And that particle then can be coupled onto the inorganic particles. And as I'll show you, we initially just wanted to have a central inorganic particle onto which was coupled organic particles. And what we found instead was that there was multiple coupling reactions occurring between not only the organic particles onto the inorganic, but also the organic particles to the organic particles. And that's what creates this network-like structure. If alternatively we take the block hole polymer that is designed for supramolecular assembly and don't assemble it and stabilize it and then couple it to the inorganic particles. Instead, if we co-assemble it together with the inorganic particles, then during the assembly process, those inorganic particles can be trapped inside, and then stabilization reactions can be performed to generate what we're calling MSCKs. And so we're going to start with these materials as the first that we elaborate upon. Now, Adriana Pavia-Sanders was a young PhD student in my group when the Deepwater Horizon oil spill occurred, and she got excited about trying to build these nanostructures to address, again, that challenge. And so, they were designed in such a way as to have inorganic magnetic nanoparticles trapped within a polymer framework, where that polymer framework would have hydrophobic polymer chains within a core domain surrounded by hydrophilic polymer chains that are then cross-linked together to stabilize the entire entity as essentially a nanoscopic single molecule containing these inorganic particles. So Adriana began with some simple polymers that we had in the laboratory, some block hole polymers based on polyacrylic acid connected to polystyrene. So polyacrylic acid is hydrophilic, it's water soluble. Polystyrene is basically styrofoam and is water insoluble, as you know. And so when these two polymers are connected to each other, whereas they normally would want to separate from one another, with the polyacrylic acid, say, dissolving in water and the polystyrene precipitating, instead, the polystyrene precipitates but is surrounded by intimate contact with the polyacrylic acid, and that allows for this multimolecular assembly into micelles to generate nanoscopic assembled materials. And when this assembly is done in the presence of inorganic iron oxide nanoparticles, those inorganic iron oxide nanoparticles get trapped within that polymer micelle. And so we do this by starting with organic solvents and then transitioning over to water, and ultimately these micelles are suspended within an aqueous solution. We then add, say, diamines and a carbodiamide activating agent to form amide bonds, where we consume a fraction of these acrylic acid groups, and that then stabilizes this structure. So again, this was the overall design. This is what we wanted to make, and it was remarkable when Adriana then collected the transmission electron microscopy images. She found first that the particles were fairly uniform in their sizes, and when she zoomed in and, say, evaluated a single particle, you can see the zoomed in image here illustrates heterogeneity and contrast. And that heterogeneity is due to the heterogeneity in the presence of the inorganic particles, which don't allow for electrons to be transmitted, and organic material that's not so good at blocking the electron transmission. And so we see these dark and light regions. Now, this told us that there was a combination of polymers and particles, but we didn't know whether the inorganic particles were on the outside of the micellar assembly, on the inside, or a combination of both. And that then was revealed by doing cryogenic tomography imaging. And you can see that in this reconstructed image that the inorganic particles are in fact embedded within the organic polymer framework, as is illustrated in the upper right in the schematic representation. So these particles, in terms of their hydrodynamic diameter, are about 80 nanometers in diameter in terms of the number average hydrodynamic diameter as measured by dynamic light scattering. In the dry state, they're about 70 nanometers in diameter. So they're quite small, uniform particles. And when Adriana tested how well they could perform in collecting crude oil, she conducted a series of experiments. And what's shown here is a photographic image of 3 vials, each containing water with crude oil floating on the surface. And then there's just going to be a cartoon representation of her administering the nanoparticles. But after a period of just minutes, when you see the image of those vials, now the crude oil has all been collected within those nanoparticles. The nanoparticles start out as kind of a light brown color, and you can see here these are quite dark in color. So the oil-filled particles are darken in color. They also then float and aggregate together. And so you can see these aggregated floating islands of particles on the surface of the water. And then when a magnet is placed next to the vial, those particles then migrate to the vial wall, and then the residual solution can be decanted and then extracted for chromatographic analysis of the remaining crude oil that was not collected by the particles. And so in the analyses here, let me just go through this quickly. Adriana always used an excess of crude oil. So she's using from about 3 milligrams of crude oil per milligram of nanoparticle up to almost 17 milligrams of crude oil per milligram of particle. And you can see that in all of these cases, she's able to collect about 80% of the crude oil until she exceeds up here in about, 16.8 milligrams of crude oil per milligram of particles. And then it drops to maybe about a 55 to 60% recovery. But at that point, she's recovering 10 milligrams of crude oil per milligram of particle. So this was a remarkable finding that they could take up 10 times their mass in crude oil. What we also found is that when she went to really high loadings of crude oil relative to particles, the system got to the point where this oil could be removed by a pump, for instance. It's no longer just a sheen on the surface of the water. And what was also found was that the crude oil that we were working with, even though it had been weathered, was still a complex mixture of hydrocarbons. And that's seen by the GC mass spec trace here. And then after the crude oil had been captured by the particles, what we find is that the GC mass spec trace basically overlays. So even though these particles are simple, they're comprised of polystyrene, polyacrylic acid, cross-linked through these triethylene glycol diamino cross-linkers, they can capture this complex mixture of hydrocarbons. Okay, so then we started thinking, well, what other pollutants are out there that are difficult to recover from the environment? And one of those that you may be familiar with is perfluorooctanoic acid. And perfluorooctanoic acid is persistent in the environment. It also persists in animals and in humans. And it persists by binding to, say, serum albumin and other serum proteins. It also persists by forming micellar assemblies because it has this hydrophilic head group and a hydrophobic tail. And it's a fluorocarbon, so it's chemically robust as well. So Adriana had said, well, let's take these magnetic shell cross-linked Knedel-like nanoparticles, and now instead of just having polystyrene, let's put some fluorocarbon into the core and see if we can pick up the perfluorooctanoic acid through a combination of fluorophobic and hydrophobic effects in the core domain. And she also then wanted to tune the chemistry of the shell to not only be polyacrylic acid, but maybe look into putting some amino groups in the shell that could interact electrostatically with the carboxylic acid head group of the perfluorooctanoic acid. So let me just point out again, one of the problems with perfluorooctanoic acid is that it is a health concern, and perfluorooctanoic acid has been used at various sites throughout the US and around the world, particularly at sites where fluorocarbon materials were processed, for instance, Teflon. And this is an article from nearly a year ago, but it's really saying that there is this huge problem. And if I zoom in here, perfluorooctanoic acid is also called C8, and C8 is considered to be toxic, and yet even though it was known to be toxic, it has been discharged into the environment, again, at these manufacturing sites. And now it's linked to several diseases. It says 6 diseases here, including testicular and kidney cancers. And it's also been found in the bloodstream of 99% of Americans. Okay? And so when we look at the statistics, again, out of every 20 children tested, 19 had perfluorooctanoic acid circulating in their bloodstream. So the real question is, what is the long-term health impact of this compound? So again, coming back to the chemistry, Adriana had said, well, the polyacrylic acid block polystyrene material works so well with the crude oil cleanup. Let's just throw a little fluorocarbon tail on a fraction of the styrenyl repeat units along that hydrophobic block segment of that, what was a diblock polymer structure. And then again, replacing this carboxylic acid group with an amino group, she looked at an amino ethyl acrylamide as the hydrophilic block segment and replacing the polyacrylic acid to see how the influence of the chemistry of the shell would play in the ultimate performance of the materials. So she made a series of these polymers having various relative lengths of the hydrophobic block segment relative to the hydrophilic in each case, where she had, say, a relative value of 100 repeat units to 25, or doubling that to 200 in each of these cases. And then she assembled each of these into nanoparticles together with the iron oxide nanoparticles, cross-linked the shells, and then evaluated their ability to capture perfluorooctanoic acid from an aqueous D2O solution where she could use fluorine-19 NMR spectroscopy for quantification. And what she found is that for the two polymer materials that have the polyacrylic acid segment, in one case she was able to recover 25% of perfluorooctanoic acid. In the other case, 35%. In the cases where the amino group was present in the shell, we thought those were going to be better at capturing the perfluorooctanoic acid. They actually turned out to be less able to capture the perfluorooctanoic acid. Here only 13%. In this case, about 10%. And so right now, we have not published these results yet because we're still trying to figure out what are the parameters that lead to the ability of these particles to capture perfluorooctanoic acid. And our current hypothesis is that because these two particles were larger in dimensions, 60 and 72 nanometers in diameter respectively, versus 40 and 42 nanometers in diameter here, that may have impacted the ability to package the perfluorooctanoic acid. So even though these numbers don't look great, when I say 10%, she was using a 1-to-1 weight-to-weight balance. And so that's still taking up, say, 0.1 milligrams of perfluorooctanoic acid per milligram of our material. And so at the best case, where we were able to take up 35%, that's 0.35 milligrams of perfluorooctanoic acid per milligram of these magnetic shell cross-linked Knedel-like nanoparticles, and that's still 2 orders of magnitude better than comparable nanomaterials. Okay, so then let me turn to the second morphology for these materials, and that's the work of Jenny Flores. And Jenny was interested in being able to, say, access the cleanup of pollutants in an environment where there might be complex mixtures of pollutants. And that's why we came up with this kind of a design strategy, where Jenny said, well, maybe one of these particles should be optimized to take up crude oil, and maybe another one should be optimized to take up perfluorooctanoic acid, and maybe a third would pick up arsenic or mercury or something. So we could have individual organic particles optimized for particular pollutants, and then bring them together into a composite structure where there would be a central magnetic nanoparticle onto which these were connected. Well, this was actually partly my design, and that's the failure of it, as I'll show you in just a moment, was partly due to the fact that we didn't understand that as nanoparticles get larger, the inorganic nanoparticles, they become permanent magnets. So when Jenny started this project, she was working with large inorganic iron oxide nanoparticles, and she found that they just kept clumping together because they were basically permanent magnets. And so she scaled back on the size of those particles, and she assembled this shell cross-linked Knedel-like nanoparticle without any iron oxide, and then was interested in coupling this organic particle onto the little small inorganic particles. And so let me show you what her result was here, where all she had to do was basically take the same kind of block-copolymer that Adriana had been working with and assemble it into Micelles in the absence of any inorganic nanoparticles, and then conduct the cross-linking in the shell. And then she could then take an inorganic iron oxide nanoparticle that now presents amino groups from the surface, and then she could take some of the residual carboxylic acids here and do a coupling reaction to establish an amide linkage. And what she found was that in the first step, the cross-linking reaction involved the addition of a carbodiamide to activate carboxylic acids. And so if those activated carboxylic acids, they form these activated esters, they encounter an amine, it forms an amide bond. If they don't encounter an amine, they get hydrolyzed, and the activated ester gets broken down. And so in the first stage of the reaction, the cross-linking occurred intramicellarly. But then when she had those cross-linked micelles and she added more carbodiamide in this second step, what she found was that some of these amide linkages that had formed might have a dangling amino group on them. So the shell of this particle isn't just presenting carboxylic acids, it's also presenting amines. And so that allows for coupling of one organic particle to another organic particle, because an amine from one particle combined to a carboxylic acid on another one. And she figured this out initially by looking at transmission electron microscopy images. On the left side here, you see this TEM image shows us down in this region that there's this heterogeneity of contrast. There are some dark regions of inorganic particles not allowing transmission of the electrons, and there's some lighter regions of the organic particles. But up in this region, you see there are organic particles clustered together without any inorganic particles linking them together. And so we weren't sure whether that was just a drying effect on prepping the sample on the TEM grid. So we engaged in a collaboration with Darren Pochan at the University of Delaware, and his PhD student, Ying-Chao Chen, who later became a postdoc with me, conducted the cryo-EM imaging and found that, in fact, in solution, the organic particles are indeed coupled to one another. Now, Jenny did a bunch of other experiments to confirm that this was the case, and then moved forward with testing how well these could perform in collecting crude oil from aqueous solutions. And here you can see some actual images of the lyophilized powder of these magnetic hybrid network materials as Jenny dropped it into the vial. And then after a period of time, you can see that she's collected the oil-filled particles, and there's some crude oil here that you may be able to still see floating on the surface. And then she analyzed the residual solution for the amount of crude oil that had not been captured by these magnetic hybrid networks for their quantification. And as you can see here, the max loading capacity was about 3.5 to 4.5 milligrams of crude oil per milligram of these magnetic hybrid networks. So they're about 1/3 to 1/2 as efficient in capturing the crude oil as Adriana's particles had been. But what Jenny further did was she confirmed that she could recover the material by the magnetic effect. Because if there are organic particles coupled to organic particles, we were concerned that they may not have an inorganic particle connected to them, then they wouldn't be collected by when the magnet is used. And so what she found is that she could, in fact, recover about 90 to 93% of the particles. So 93% after deployment, and then 90% after organic wash to extract the crude oil out of the particles to allow them to be recycled. And when she recycled them, then she found that in a first pass, they were able to capture a certain percentage of the crude oil, and they performed about the same in at least 3 iterations. So they are recyclable materials. So then Jenny was interested in increasing the amount of crude oil that could be captured by her materials. And she also was interested in addressing questions that we were receiving from people about, well, you know, you've shown how they perform in water, but how about salt water? How well will they perform, say, in the Gulf of Mexico or in a saltwater environment? And so what she did was she backed off from studies on crude oil, instead was working with dodecane as a model hydrocarbon material. And what she found was that she was shaking the samples and even vortexing them to try to get more dodecane to be captured by these materials. And ultimately, she started seeing these emulsions forming. And the emulsions could form in water, but as you know, when you're in the laboratory and you have an emulsion, you add brine to salt things out, right, and to break up the emulsion. And so in the saltwater condition, she was not able to emulsify the sample, and so she reached a peak for the maximum dodecane uptake by these materials, likely through sequestration into the hydrophobic cores of these materials, whereas in an aqueous phase, she could capture the crude oil not only through uptake into the particles, but also through this emulsification effect. And so we wanted to confirm whether these particles were actually acting as particles in the formation of a Pickering emulsion. And Pickering emulsions are quite common for particles, but Jenny's particles are unusual in their shape, right? They're not just a simple particle, they're rather this network of particles. And so what she did was she labeled the particles in order to track where they were residing, and then Soon-Mi Lim performed microscopy measurements and also confocal fluorescence microscopy experiments, and what we confirmed was that the particles are actually at the interface between the oil and the water, so they are acting as Pickering emulsifiers. Okay, so I want to show you a quick video of these materials because they're actually quite fun to work with, and we've developed a youth adventure program for high school students to come in the laboratory and make these materials and do some experiments with them. And you can see how magnetically responsive they are. These emulsions, you can basically move around a vial in real time. And so we've talked with ExxonMobil and some other companies down in the Gulf region about potentially using these materials, say, in assisting in cleaning up water from fracking processes and other activities. Okay, so I've made these materials sound like they're really great, right? So now we have to get into, what's the problem, right? And we come back to this block polymer structure that's used to assemble into these materials. And as I said, it's comprised of polyacrylic acid connected to polystyrene. And I mentioned that polystyrene is styrofoam. I didn't mention that polyacrylic acid is actually the super absorbent hydrogel material in baby diapers, so considered to be safe. Styrofoam is considered to be safe, but the problem with both of these is that they're persistent in the environment. They don't undergo breakdown. They're also petrochemically sourced in terms of the monomer building blocks from which they're produced. So two issues that your father would actually not be satisfied with, right, in terms of being able to recycle and have address issues with sustainability. And so you may have been hearing in the news lots of issues related to plastic materials and their persistence in the environment. There is this place called the Great Pacific Garbage Patch. It's estimated to be 3 times the size of France, having something like 1.8 trillion pieces of plastic in a mass of 79,000 tons of plastic debris. And it's just basically floating between California and Hawaii. So that's one problem. Here's a Facebook post from one of my friends, Anna Malcock, when she was on holidays last summer with her family, and they'd gone to Malta, and she says, "It looks beautiful from a distance, but the blue lagoon in Malta should be renamed the trash lagoon. My kids would not even go into the water due to all the plastic waste everywhere. We need to solve this immediately." And you can't see the images, but there's a bunch of plastic floating around and just left as debris. And then those are cases where we can see the plastic. We know there's a problem. There are other issues with plastic in the environment, including things like microbeads. So you may have heard about microbeads. They've been used in things like cosmetics and face scrubs, toothpastes, et cetera, lots of different products. And now they're starting to be banned as well because they may be able to harm wildlife and even people. And in fact, more recently on the news, I don't know if you've been hearing about the sea turtles that they're finding, the baby sea turtles that are dying, and they're finding them with microbeads in them as well. And so it's a real issue that we have to think about. So for probably the first 10 years of my career, my primary goal was to take small molecule monomers that are held together through relatively weak intermolecular interactions and polymerize them and make long chain structures or various architectures so they can tangle up and they can exhibit the kinds of properties physical and mechanical properties that we think of in terms of plastics, right? And so I wanted to make polymers for functional performance. And usually I think, well, you know, polystyrene, it's benign, polyacrylic acid, it's benign, so if it's lost in the environment, it won't really be a big deal. But the problem is that lots of polymers that you think are never going to break down actually can. And so we have to be thinking about what are the degradation products from these materials. And those degradation products should be biologically and environmentally benign. And actually, we'd like them to be beneficial. So our strategy over the last probably 10 years has to really think about what kinds of monomers we use as building blocks. We want those monomers to come from natural products, so be renewable, so they're not petrochemically sourced per se, but not necessarily for that as our key motivation. Our key motivation is that once we make a functional material, we want it to be able to degrade back to those starting materials after it's served its purpose. And so that's, again, the overall life cycle. But I want to show you one example of a polymer, you know, that was an engineering plastic that was never meant to undergo degradation, and that's bisphenol A polycarbonate. It's a fantastic engineering plastic. It's a great material. It's hydrolytically stable until it's under extreme conditions. So when this is placed, say, in a dishwasher under the temperature and the pressure and the water, then it can undergo some degree of hydrolysis and release bisphenol A. And bisphenol A has been implicated in cancer and other diseases. It's basically like a hormone disruptor, and so it has issues associated with it. And so I just want to take a really quick tangent, since we're so short on time, and tell you that we've been looking into nature for alternative phenolic natural products that can be used in place of bisphenol A. And one of my students identified that there are these compounds that come, in this case, from magnolia trees that are neolignin-based structures. So you may have heard the word lignin with an I. This is lignan. A lignan just has a beta-beta linkage, and a neolignin has a non-beta-beta linkage. And in particular, my student, Kevin Wacker, identified two neolignans that come from magnolia source, and these are hinokiol and magnolol. And what's really beautiful about these phenolic compounds is that they have two phenols, so we can make a polycarbonate chain, but they also have two alkenyl groups on each one of their repeat units. So they have this orthogonal reactivity. So they can be obtained on kilogram scale. They've been studied for therapeutic applications. And again, these diverse functionalities allow for us to produce polymers and also chemically modify them. Again, back to Marino's interest in, say, reactive processing of materials. So this is a picture of Kevin, and this was his roadmap for his PhD, and he actually just finished a few months ago. What he said was, well, from hinokiol and magnolol, he could go to polycarbonates, like we were initially targeting, or he could go and make some thermosets. He could make some polyolefins by some, say, metathesis chemistry. So this was his chemistry, and I was told don't show too much chemistry. But let me just focus on this small area down here in thermosets. And even from one small molecule, magnolol, he could take magnolol and convert it into a lot of different kinds of monomers that could be studied for their cross-linking reactions in thiolene, photo-initiated thiolene chemistry to make cross-linked networks. And these cross-linked networks are actually lovely materials. The chemistry can be done without solvent. There can be a diversity of properties depending upon the chemistry that's incorporated here. And we published some of Kevin's work and just recently engaged in a collaboration with Andrew Dove's laboratory, who was at Warwick and has recently moved to the University of Birmingham. And a postdoc in his group, Andrew Weems, had been a PhD student at Texas A&M University and collaborated with my lab. So it's perfect. We've got Andrew over in Andrew's lab doing 3D printing of some of these monomers from magnolol and also oligomers and polymers that are polycarbonates of hinokiol, and building up interesting devices for cardiovascular but other directions that we're going to be pursuing. Okay, so let me come back to this block polymer structure, because I need to make a full circle back to the nanoparticles for the environmental cleanup. And just say that the work that Kevin had been doing would allow us to get to polymers or cross-linked networks but in an uncontrolled kind of condensation polymerization. But in order to make well-defined nanoparticles, we need to have this amphiphilic block structure. So we need to have a hydrophilic block and a hydrophobic block connected to each other. And we need to define how long each of those is, because that controls the size and the shape of the assembly that they make when they're put into water. So we needed some chemistry to take natural products and build them into these amphiphilic block polymer structures. And that chemistry was developed by Koichiro Mikami, who was a postdoc in the lab. And again, this is a lot of chemistry here. Let me say that the key starting material is this cyclic carbonate of glucose. So he can start with a commercially available small molecule, do a couple of chemical reactions, and access this species that then has, what's important here is this 6-membered carbonate ring, because in the presence of an alcohol and an organobase, this can undergo ring opening polymerization, and that's a chain growth polymerization, and it can be well controlled. And Koichiro showed that he can control the degree of polymerization. By MALDI-TOF mass spectrometry, he also confirmed that each of the polymers, and we have different polymers of different degrees of polymerization here, where we have one with a degree of polymerization of 21, 22, 23, 24, 25. So we still get a distribution of molecular masses and degrees of polymerization, but each one of those species has the initiating end at the alpha chain terminus and the terminating moiety at the omega chain end. And what that means is that the initiator initiated the polymerization, the chain propagated, and then we were able to terminate it. And by that process, then, that allows us to get into growing block polymers, because we can initiate, grow a segment, then once that monomer is consumed, add another monomer, and then initiate and propagate that monomer so we can build these block polymer structures. And so that was done by Lu Su, and she recognized that Koichiro had been working on just this one monomer, but all of these species have that 6-membered cyclic carbonate, and they just have different things dangling along the side. And those dangling groups are critical to the behavior of the polymers. So 2 of them that she's focused on are this one, where she has ethyl carbonate groups as what are protecting groups, but they're also labile. So this monomer and polymers from it will break down into methyl glucose, which ultimately will lead to glucose, carbon dioxide, and ethanol. And she's also focused on this second monomer, which has not only an ethyl carbonate at this, what's called the 2 position, but also a propargyl carbonate at the 3 position. So now that brings in side chain functionality. So from one particular block polymer that has, again, the ethyl carbonate protecting groups on this segment of the chain, and then an ethyl and a propargyl carbonate on this end, we can take that triple bond and do chemistry on it. And so that chemistry can involve, say, reaction with an azide, so an alkyne azide click reaction, or reaction with a thiol and a thioline reaction. And that can bring on to the side chain hydrophilic moieties. And those can be neutral, like a polyethylene oxide. They can be cationic, they can be anionic, they can be zwitterionic, etc. So there's a great diversity in the kinds of polymers. And then each of those polymers now has this hydrophobic segment here. These are all hydrophobic. And then a hydrophilic segment, where the hydrophilic segment is different in its composition. And so these can be supramolecularly assembled into micellar structures in water. And in fact, for these 2 dye blocks, they can be directly dissolved in water to form well-defined micelles. In this case, this hydrophilicity of this segment wasn't sufficiently high, so this block polymer required transition from organic solvent to water, and it formed vesicles as opposed to core shell micelles. Okay, so I think I may have, about 5 minutes left or so? Okay, I focused on our environmental direction in our application and cleaning up pollutants in the environment, but another huge area in my research group is making well-defined nanoparticles that are designed for drug delivery. And we've been working in this area since the late 1990s, and through many different biodistribution studies and pharmacokinetic studies, et cetera, with intravenous administration and systemic delivery, we've found that we just can't overcome the problem that most nanoparticles, when they're put into the bloodstream, end up in the liver, so 80 to 90%. And so what we've done over the last few years is we've recognized that an alternative approach is to instead deliver directly to where the disease is located, right? And so we're targeting recurrent urinary tract infections in one case because we can access the urinary tract through a catheter. And we're doing a lot of work in the lung, and we're looking to treat lung infections and also metastatic cancers in the lung, especially those that arise from osteosarcoma. So the overall idea is that we take these well-defined particles that might carry imaging agents and pharmaceutical agents and deliver them by inhalation directly into the lung. And again, we're building these particles now from using glucose and other sugars as the building blocks. And I just want to show you one aspect, and that's treating osteosarcoma lung metastases. So from the polymers that Lou had produced, she looked into loading paclitaxel into the particles. And what she found was that she could take this block copolymer and co-assemble it with paclitaxel and then result in paclitaxel-loaded micelles, or she could take this block polymer, assemble it, cross-link it, and then co-assemble it with the paclitaxel. And she could also then load this into what is a shell cross-linked version. And she could get about 10,000 times greater paclitaxel concentration in aqueous solution versus paclitaxel when it has to be dissolved. So these particles are able to package it within their core. The problem with these materials is that when they were then put into a sink environment, the paclitaxel would leak out. So we couldn't contain it indefinitely and trigger its release. And so what we've done more recently, and this work was just published earlier this year, is that we've been now taking the paclitaxel and dimerizing it. So we take two paclitaxels, dimerize them, but through a disulfide linkage in the center, and then esters to the paclitaxel. And so upon reduction of that disulfide, it releases a thiol that bites back onto the structure and kicks out paclitaxel. So we start with a dimeric paclitaxel prodrug, and it breaks down to paclitaxel as a drug. And we're loading that now into a mixture of block polymers, where some of them carry a near IR label, some of them carry cationic moieties, so we can mix together the polymers and tune the properties of the nanoparticle assembly together with the drug. And so our overall target is to have these particles, again, involving co-assembly of at least two polymers together with the dimeric paclitaxel, so that in a healthy cell with low glutathione levels, there would be a low amount of reduction, a low amount of release of the paclitaxel. In an environment where there would be higher glutathione levels, then there would be higher release. And by having some cationic moieties on the particles, we anticipated that we would be able to penetrate deeper into a tumor. Okay, so these data are just illustrating that there is some differentiation in the toxicity against cancer cells. SGSA is an osteosarcoma cell line. It's a human cell line that was isolated by Dennis Hughes' laboratory at MD Anderson. So we have a much lower IC50 against this osteosarcoma cell line than a non-cancerous cell line. And then when we look at spheroids, these particles are able to penetrate. These are the dipaclitaxel loaded materials. They can penetrate into the tumor and result in the SGSA cell killing within the tumor spheroid. And finally, in an animal study, we're using an orthotopic model where those SGSA human osteosarcoma cells are injected into the tibia of a mouse. That generates a primary tumor, and then that primary tumor continues to grow and ultimately begins generating metastatic tumors, and those metastatic tumors are predominantly in the lung. So when we deliver our particles by nebulization and the mice inhale them, then afterwards we do ex vivo experiments to see if we can see the particles. And it's difficult to see here, but there are red splotches throughout the lung tissue, indicating that the particles were able to reach virtually all lung regions. There's still quite a lot of metastatic tumor growth, however, and we've not been able to knock down the tumor growth sufficiently. We have shown that for our dipaclitaxel-loaded micelles, the data in blue, that we have less tumor burden than in the case of nebulization of just dipaclitaxel or a control of saline. So we do see a statistically significant reduction in the tumor burden, but not sufficient at this point for our satisfaction. And as one would expect, because we're delivering to the lung, we don't really have systemic delivery of the particles, and we don't have any effect on the primary tumor. Okay, so I'm just going to bring this all back full circle here in this one slide and say that now at this point, May is a current PhD student, and she's using the chemistry that Koichiro and Liu had developed and taking these glucose-based monomers, conducting ring-opening polymerizations, and developing polymer materials that can be co-assembled with iron oxide nanoparticles to make these, again, magnetic shell cross-linked nanoparticles, not based on polystyrene and polyacrylic acid, but now based upon hydrolytically degradable glucose-based polycarbonate materials, again, to feed them back into the environment. And I'm going to have to just jump to the end now. Let's see. Okay, I want to give you another take-home message and say that I think with your father's approach to recycling, I think that's really important. And more people have to be thinking about that, not only in science, but in the community. And I think the community is largely pushing scientists to be thinking about recycling. And my message with recycling is that there's a lot of recycling that goes on in the world, in the non-synthetic world. So if we think about in the animal world, animals eat other animals, so there's a recycling process. Animals eat plants, so we recycle there. Plants actually can eat animals as well. And plants and animals both eat minerals. And then there's further recycling in the geological terms as well. And so we really have to be thinking from the very beginning about building our functional materials in such a way as they can accomplish these goals where they're not persistently a problem in the environment. Okay, so these are just some of the natural products that my lab is working with to build up polymer materials. Because a lot of what we work with is sugars, we've trademarked the term sugar plastics, and I have to disclose that I actually started a company last December called Sugar Plastics LLC, if you're interested in taking a look at our website. We have almost nothing going on because there's another company licensing our technology right now, but that's a company called Taysha. And Taysha is a company that I serve on their board that's going to license the technology from Texas A&M, develop our sugar plastics for broad applications, and then my company, Sugar Plastics, is going to sub-license some of that back. That's the overall scheme. So maybe in a couple of years, I can tell you more about how that's been successful. I tried to mention the people from my group who've done the research throughout the presentation. I just would like to finally highlight the fact that the Doherty-Welch Chair in Chemistry supports a lot of the research that goes on in my group, and it allows me to seed new projects, and it's what allowed us to get into this area and start looking at sugars as building blocks for polymers. And then we have acquired funds from the National Science Foundation to develop the materials further. So the final slide, as I promised, is last night, the lottery tickets. And you see, he's quite proud of his lottery tickets, and I only got 4 numbers out of 5 picks. 4 out of 5 had one number, basically. So I don't know if anyone else did any better, but I think we should go Powerball, still on, right? Okay, so thank you so much for your welcoming.
Well, thank you, Karen. It was excellent. Really enjoyed your seminar. My question is about your work. It's really interesting with the oil and the PFOAs. And have you been investigating the stability of these particles as a function of time and water chemistry? And with these nanoparticles, these iron oxide, have you looked at possible formation, transformation, or other coatings that could form on them that could possibly impede?
Yeah, so those are excellent questions. And some of what I didn't get to talk about is we are looking into those things now. So the problem that we have is that there's just so many variables, right? And so many different things. So when we look at May's scheme here, we have to control, the length of this, the length of that, whatever the side chain groups are, whatever this thing is, whatever the proportion is with the iron oxide nanoparticles. We have to think about all of those parameters, even to make the materials and then to study their behavior. And so what we've done is we have a new DMREF, well, relatively new, it's about a year old, DMREF collaboration. And it's with Arthi Jayaraman, who does a lot of computational studies and tells us what we should be, what polymers we should be making, what inorganic particles we should be mixing with them to make these morphologies. And then the key thing that I think is related to what you're asking is Bill Johnson at University of Utah, where Bill is actually interested in doing like field studies. He does laboratory transport studies of these particles, because that's a key question, right? Not only what does the particle do in terms of taking things in, how does that particle interact with other surfaces and interfaces, and what is the transport like, right? So we're just exploring some of those things. Yeah, thank you.
Thanks, Karen. I have a question for the last part that you mentioned that using nanoparticle for the lung cancer treatment. So what's the administration way that you use the systemic administration, or what's the target mechanism?
No, it's inhalation.
Okay.
So we have a nebulizer for the mice. Yeah.
Will the particle penetrate the mucus or not?
So we get that question a lot, and we have designed some studies where we'll look at mucosal transport, but I can tell you right now, I don't think it's going to be a problem, because, and the reason I say that is because we had some early generation particles that we loaded with silver and looked at treating lung infections, and we could find that the particles loaded with the silver could be administered at 16 times less silver mass than if silver was a small molecule or a salt, and still have 60% survival. So somehow, whether the particles have to get through the mucus or whether they just hang out in the lung and release the drug and it sort of traffics in that region, I think we still have benefit. So I don't know if the whole particle has to get anywhere.
Okay, you think it's just because the smaller size?
I think it's the depot delivery. So we've labeled the particles and we've monitored their presence in the lung over a period of time. And they have a half-life of clearance from the lung of about 8 days, whereas the small molecule clears in like 0.2 days, right? So I think it's just the fact that instead of going systemically and delivering things everywhere, it's just there. And leaching out of the particle is still going to be in contact with the lung.
Yeah, I see. It's just kind of like a drug depot,
right?
Right.
Nice work. Awesome work. Are the nanoparticles scalable for use in in the deep water horizon kind of situation?
Yeah, that's a great question. So we filed an invention disclosure, and my technology transfer office told me these are going to be too expensive, and they wouldn't file the patent. I had a master's chemical engineering student who then did some calculations and found that they're actually not that bad. I mean, they're a lot more expensive than polypropylene or polyethylene, but they're less expensive than some of the catalysts that are used to break down the oil. So I think in terms of cost, I don't think they're that bad, but we're not thinking that we would put them out in the Gulf of Mexico. Rather, I'd like to deploy them at sites of water intake, right? So let's say your water intake pipe can have the magnetic system to recover them as well. So you deploy them, and then they clean up the water at the water intake sites, for drinking water or cooling water, or cases where, yeah, you don't want to be taking in contaminated water.
Perfect time. Okay. So I'll tell you. So,
Thank you.
again.
Thank you. Thank you.
Thank you.